Thank you everyone for joining us on this Thursday evening. And this is the one of the very first events that we're organizing in the new quarter, and it's titled the latest of cell therapy. And I thought to put together this event just as a reality check for where we stand in terms of cell therapy. I think there's been a lot of hypes versus real progress that has been made. So I'm very honored today to have two stellar professionals in the area of cell therapy to guide us through the conversation. But before we get into the meat of the conversation, I just want to quickly introduce myself. I'm Richard. I'm an MIT alum, class of 2021. So I just graduated a couple of months ago. I was a PhD at MIT. So shout out to anyone out there who's also a postgraduate at MIT. And I'm currently working as the director of accelerator and also the chief of staff at Pear VC. We're an early stage venture capital fund based out of Palo Alto. And I am also the healthcare lead of the MIT Alumni Club of Northern California, along with Afsana. Afsana, if you want to quickly unmute yourself and say hi.
Yes, hello. Sorry, my lighting's really bad, but yeah, very excited to be here. And as Richard mentioned, I'm Afsana Akhtar, one of the co-chairs. And yeah, I look forward to the conversation. Thanks, Richard.
Thank you, Afsana. And really the purpose and really the goal of the healthcare track is to gather the minds of MIT alum community and also beyond to have a genuine discussion about the current progress of healthcare, medicine, and biotech. And we put together all sorts of events like the panel discussions that we're having today to guide us through the discussion. And in the spirit of the events that we are organizing, I just want to quickly mention the one event that will happen. So I'm sharing my screen. Every year we put together this conference called the Future of Healthcare and Medicine Conference. It consists of a series of panels and speakers, and it's a great way for you to broaden your network and interface and with other professionals who are also interested in healthcare and medicine. There's a kickoff. So this year I'll be leading the organizational effort. We will have a kickoff session October the 2nd. If you're interested, just make sure you sign up through this website. It's actually free, whether you are a club member or not, unlike this event, which is not free if you're not a club member. So yeah, make sure you sign up. We'll have lots of fun together. And I will make sure to share this link in the chat. Okay, enough of the logistics. Let's actually get started on the panel. So today we're very honored to have Shannon and Dahl, one of the seasoned veterans in healthcare and biotech venture building, and also Eddie from Senti Bio and Pear VC. So I am going to kick off the panel by asking each of them to introduce yourselves. So Shannon, you want to give us a start?
Sure. Thanks so much for having me. It's great to be here. Looking forward to the conversation today. And yeah, I got into the space actually at my time at MIT as an undergrad in the late 90s. Tissue engineering was evolving in Boston at that time, and I got really excited about it. And it's this space of regenerative medicine where I like to say cell therapy is the raw material instead of the final product. We grow tissues from cells. Got really excited about that, went and did my PhD at Duke in biomedical engineering, and then co-founded Humacyte, which was a company where we advanced a bioengineered blood vessel from academic research through phase 3 trials, and the company actually just went public a few weeks ago. And you know, we were an early mover in the space, and one where we laid a lot of pioneering groundwork for this field, the cell and gene therapy and regen med. focused on indications in cardiovascular and the renal area. 3 years ago, I left and joined Cell Care Therapeutics, where we were developing biologics derived from an allogeneic mesenchymal stem cell platform, really focused on their secreted factors, which are responsible for their signaling to other cells, and capturing those secreted factors, which are microRNA and exosomes and proteins, to really modulate the immune system and turn down inflammation in the eye and the lung and the brain and the gut. I was serving as Chief Scientific Officer there until last month. And then I'm on advisory boards for Strand Therapeutics, which is mRNA for cancer, and Hepatics, which is a Stanford spinout for liver cell therapy. And I'm actively exploring potential CSO roles here in the Bay Area, ideally in the cell and gene therapy sector. So I'm excited to bring a broad perspective on the field at large and comment on where I see a lot of excitement for opportunities and also risks and perceived risks.
Thank you, Shannon. And Eddie, if you want to introduce yourself.
Yeah. First of all, thanks so much to the MIT Club of Northern California for hosting me in this fabulous event. Thanks to Richard and Afsana. And yeah, I'm likewise really looking forward to this discussion. I've been working in the cell and gene therapy space now for well over a decade, starting to get the gray hairs to show it. I grew up here in the San Francisco Bay Area. I pursued an undergraduate degree in chemical engineering at Stanford. And at the time, Shannon mentioned she was super interested in tissue engineering as that was emerging. At the time that I was, thinking about pursuing graduate studies, to me, the explosion of interest in biotech, broadly, not only cell and gene therapy, but also genomics and systems biology and synbio. Those were the things that I was really excited about, and that led me to MIT to pursue a PhD in biological engineering, course 20. And there I somehow, found my way to Bob Linger's group, was one of the many in his lab, and was fortunate to take me on. I conducted my PhD research in his lab as well as Dan Anderson's, and there I worked on non-viral gene therapies, had a terrific group of collaborators, got to synthesize and screen really large libraries of polymer and lipid-based materials for the delivery of macromolecules, including DNA, RNA, proteins, and all sorts of applications, cancer and heart disease among them. 10 years ago, I used to have to really explain and stretch to tell people what is lipid or polymer-based delivery of nucleic acids. And I feel fortunate, and it's, we don't often talk about miracles in science, but it's kind of a miracle, as a scientific person that, we now have scaled up that technology, and, well over 100 million people have been dosed with a lipid and RNA at this point. So after wrapping up the PhD, I joined a company that Bob Langer had co-founded called Kickoff Pharmaceuticals. It was a bit of a departure. I focused there on novel formulations of drugs for ocular diseases, and worked in R&D for 2 years there. And while I was there, I really got to understand how to innovative biotech companies, like the one I was at, develop their business model and strategy. And to pursue that and scratch that itch, I went back to Stanford for an MBA. For my summer internship, I got to intern in business development at Genentech and Roche. And then actually, got introduced via the VC firm that Richard is at, Pear, to an MIT spinout out of Tim Liu's lab at MIT, and it was called Senti Biosciences. And Tim was serving as the CEO and co-founder. We got connected and really hit it off. I joined as the first business hire about 4 years ago now, and since the time that I've been there, it's been a really fantastic ride. I've been leading corporate and business development there for the last 4 years. And to say a little bit about Senti Biosciences, it's a synthetic biology based company that's trying to engineer next generation functionality into cell and gene therapies. This year has been a really exciting one for the company. You know, we had a Series B raise at the start of the year. You know, we have a bunch of data disclosures at some of the industry conferences for our allogeneic CAR NK cell programs for cancer. And I was fortunate to work with my team to close 2 biopharma collaborations earlier this year. One was Spark Therapeutics in the area of in vivo gene therapies, and a second with Blue Rock Therapeutics for regenerative medicine. And recently, in addition to my full-time role at Senti, I've had the pleasure of working with Richard at Pear as an advisor to its life sciences investments. On the personal front, I live here in the Bay Area on the peninsula with my wife and son. Son is almost 2 years old. You might hear him in the background. It's dinner time. We're expecting actually another baby, another son in the next week. So I hope I can stay on for the whole thing. I'm going to be checking my phone, but it's a pleasure to be here.
Yeah, thank you very much for the awesome and detailed description, Shannon and Eddie, and congratulations on your wife expecting. And if your son ever wants to join us, I'm sure we'll receive him very, very well.
Thank you.
So I think both of you came from very impressive and amazing backgrounds, and it seems to me Shannon coming from more of a regenerative medicine background, and Eddie coming from a more of an immuno-oncology cancer background. And my next question will, I'll give it to both of you, and so you can point things out from different perspectives. Our audience comes from a variety of different backgrounds, and so just to clear the air and accommodate everyone in the room, exactly what is cell therapy, and what are the different types of it? And so I'll hand this question first to Shannon.
Yeah, I think it's a good question, and there's a broad sector of affiliated technologies. If you go to the FDA website for approved therapies and cell and gene therapy, I think that kind of covers the umbrella. The field really started with cord blood cell therapies and cord blood transplants, and so those were the early approvals, and then we migrated to include gene-edited cell therapy, gene therapy, and also engineered tissues. So I think the field includes those types of products broadly. I think there's other ways to look at it too when we think about the emergence of the field. There's autologous products, so come from the patient themselves. There's allogeneic products where a donor cell population can be used to manufacture at scale for reduced cost of goods and increased consistency. There's this gene modified cell therapy where you can tune the cell to do something extra specific in a potent way. There's stem cells and differentiated cells and immune cells. So yeah, I see it as encompassing all of those types of technologies.
Yeah. Thank you, Shannon. And Eddie, anything to add?
Yeah, that was a really good description, Shannon. Yeah, she's right. It's a very broad set of therapies. Maybe just to, and I'm sure we'll dig into like lots of different subtopics that Shannon mentioned, but I think in terms of just thinking about the broad medical landscape, one thing that's really stuck out in me from the business development side, like when I work with pharma companies, they're thinking about every type of medicine out there for patients. And patients at the end of the day kind of don't really care, like what is the type of medicine that they're being treated with. They care about, is this effective? Is it safe? Is it convenient? But that said, there is just something I find incredibly exciting, and thrilling about cell therapies in contrast to all the other types of medicines. And that's all the other types of medicines. No offense, I've worked on some of them like small molecule drugs and nucleic acid based therapies, but they're really just like biochemicals. And as we all know, there's something unique about cell therapies in that they're living medicines. And the types of therapies that Shannon works on, maybe even more complex than the ones that we work on in Senti, in the sense that these are like networks of cells So I think that's kind of one of the organizing principles of the field. And Shannon mentions that there's a long history of kind of use of these cell therapies. Like, although now there's been so much money and and interest that's poured in, you can go back to the cord blood, as kind of being one of the early examples of cell therapies. Cord blood derived HSC transplants. They're really useful for a lot of bloodborne diseases. But what I find kind of really paradigm shifting about kind of going to a cell therapy is something that our founder at Senti, again, who's an MIT professor, Tim Liu, he always kind of talks about cell therapies as being essentially like mini computers, in the sense that you have DNA that you can program with algorithms, just like software. And then there's, the RNA and protein hardware that can execute those algorithms. So only with the techniques that have been made possible kind of in the past 30 years, genetic engineering, we're getting better in our ability to program these cell therapies so that they can sense different types of inputs, like, biomarkers of disease. They can compute an appropriate response and then actually execute upon that algorithm in a therapeutically optimal fashion. So kind of touching on the theme of tonight's event, like hype or, what is the reality of cell therapies? Day to day, I'm consumed in the challenges and, how do we make these things better and what are the roadblocks? But I'm really a long-term optimist with respect to, there's just something very sci-fi about the way that these medicines can work using techniques that we're now getting better at. And it's a little bit like, I think there was a 1960s movie, Fantastic Voyage, where, there was a submarine crew that shrunk down to a microscopic size and they could go into the body of a person and, repair damage. And, just the prospect that was science fiction. It's not really science fiction today, that there are cell therapies that we're able to kind of do something similar. That I just find really compelling.
I'll just build on that for a minute too. I like everything you said, Eddie. And, I think what it continues to excite me is that it's really a biologic solution to biologic problems, right? The body is complex. There's a lot going on. We don't understand it all yet. And, it seems really compelling to have a biologic response or a biologic solution instead of, a more chemical type of solution or a device-oriented solution, and for many types of diseases. So I think that's really exciting that we can more mimic what should be happening in a healthy response.
Yeah, absolutely. I think among all the promises and the optimism that cell therapy holds, and all the billions of dollars that get poured into R&D, only a handful of cell therapies have actually been approved by the FDA. There are probably up to 8,000 of therapies that are currently in the FDA pipeline. So there are a few folds to my question here. The first one is, what are some of the biggest challenges of cell therapy research and delivery? I'll give the question to Eddie.
Yeah, and I think I'm again probably going to answer just because we said the cell therapy field is so broad. I think there are some unique challenges, and I'll try to kind of get a little bit more granular and peel the onion a bit of what I view as the key challenges specifically within the cell therapy for cancer space, which is now kind of grown to be a major portion of the cell therapy kind of overall space. So in that space, I think the challenge right now is, it's common to other fields within biology, but we just have a very limited understanding of what are all the factors out there that make a cell therapy effective and safe. So to kind of dive into it, you think about this cell therapy that we're taking a cell from the body, we're reprogramming it, often in the lab, modifying its DNA to be able to target and hunt down cancer, to distinguish the cancer cell from a healthy cell, and to do all that in a way that's safe, cheap, and kind of broadly accessible for the patient. So those are kind of that's the goal that we're trying to achieve. And every single one part of that, problem, that the components of that, the field is really in its infancy. And so issue number one might be, okay, we take a cell from the body. Well, which cell? There's, as if anyone has seen kind of from those biology textbooks, the picture of the hematopoietic stem cell, which is kind of the multipotent progenitor cell that forms all of this immune cells and all of the blood and bone marrow. There's just a complex, network of so many different flavors of cells. And so, there's some key players, and, T cells have been, kind of long known as one of the key players in the adaptive immune system. And the first generation of cancer cell therapies have been based on specifically alpha beta T cells. And, even more diving more in, CD8 or killer T cells, in addition to CD4 or helper T cells. But there's all other sorts of players in this cast of characters. So there's other types of T cells like gamma delta T cells. The company that I work with, Senti Biosciences, they're working on natural killer cells, which are an innate immune player. And there's also myeloid cells, monocytes and macrophages. And at the end of the day, each of these has advantages and disadvantages. And right now, one can envision that the 21st century physician will eventually have this like armamentarium of here's what we can use and kind of pool to solve the patient's specific needs. So that's kind of issue number one, like what cell. I think the second is targeting. Like, how do you actually discriminate and safely kill all of the tumor cells, including any of the tumor cells that are kind of sneaky and that look a lot like healthy cells and can replenish the tumor. And so targeting, like, again, it's like an onion. It's how do you actually pick what is the right target on the cancer cell? It could be a cell surface marker. It could be something that's mutated within the cancer that's presented on the cell surface. If you actually target and just go after one, marker on the cell, cancer, as we all know, kind of has ways of evading or evolving, and there may be some cells that don't express that marker and they grow out and the cancer relapses. So that's kind of a known problem, antigen escape. And then the other problem is, again, a lot of those targets may be present on healthy cells, and you may do a lot of damage to healthy cells if you try to kind of hit them over the head too hard. And then on a macro level, like, how do you actually get the cells physically to the tumor? Like in the case of a solid tumor, do you administer the cells via the blood, intravenously, via systemic infusion, which is often fairly convenient and readily doable, or do you go with like a local approach, or do you engineer the cells to have some type of ability to actually migrate and find the cancer? And then on a molecular level, like, how do you actually tickle the immune cell to recognize that tumor antigen and then kind of get activated? So that's issue number two. It's, again, huge issue. Issue number three is really keeping the cells alive, active, and healthy in the patient's body. And this has been like a key problem with many of the first generation cell therapies, that they can have very amazing responses initially, but then they eventually kind of peter out and the cancer tends to relapse. And there's a couple of like very descriptive words that the field uses to kind of characterize these problems. One is called exhaustion. So the immune cells literally, like we do, if we get too much exertion, they can get exhausted, and that happens. And it's kind of a poorly understood biological problem right now. Second is that the immune system can actually be co-opted by the cancer, and the cancer has ways of shutting down these immune cell therapies. So that's kind of the third. And then, we always talk about safety issues of this first generation of particularly T cell therapies have been associated with life-threatening side effects called cytokine release syndrome, and which is essentially like an extremely bad flu, and can be very severe, and then neurotoxicity. And then lastly, how do you actually kind of produce all these therapies in a scalable and efficient way to reach all patients of all demographics without financial toxicity, which is what the field kind of talks about a lot. But in my mind, every single one of these is like a complex, optimization problem of I think we mostly know what all the levers are, and the field is, in my mind, it's almost like we're running the worst possible design of experiment study of one company is doing this cell type, this other company is doing another target, another company is trying kind of a different administration route or different indication, or so we're learning, but these things take time. And what would really help, as it with all kind of drugs, is if we had better ways of understanding how do drugs translate to the clinic. That to me is kind of the key challenge.
Yeah, thank you very much, Eddie, for such descriptive and informational information relay to all of us. So what I'm hearing is the complexity of different players is like picking a needle out of a haystack, the identification of the right biomarkers, the delivery and side effect and manufacturing scale up. I would love to talk about manufacturing scale up later on. But before I do that, Shannon, do you have anything to add?
Yeah, I think one thing just to reinforce what Eddie said about the immune system, I think we're at a really interesting time right now because there's been a lot of advances in the field of immunology. And that I think is enabling kind of the cell therapy, cell and gene therapy space, which has also been advancing right in parallel with immunology. And now they're kind of coming together. And I think to your point, Eddie, of different companies are trying different cell types. Broadly, there are diseases that we think an innate immune cell approach could be helpful, and people might be using different cell types across the innate immune system. And then there are areas where an adaptive immune cell approach might be helpful, and we're using different cell types across the adaptive immune system. So we see T cells and B cells on the immune side, the adaptive immune side, and Tregs and NK cells and others on the innate immune side, MSCs. And so I think kind of thinking about it from that lens of cell therapy and immunology are emerging together opens a lot of opportunity. And then I think we really need to focus in on what is the problem we're trying to solve and how do we maximize for potency. So as a field where, we're working out a lot of technical challenges still of, how do we, how do we best control the effect of the cells? How do we modify the cells? Maybe we're CRISPRing or gene editing or gene modifying in some way. Maybe we're using autologous, maybe we're using allogeneic. But as we work out those technical issues, I think an area that's really important for us to focus on in parallel is not just think about our biology target. What would this cell look like in health? It would have these markers and not have those markers. That's important, and we should be doing that. But we should be doing something else in parallel. And that is really having like a start from the end and work backwards perspective. What is the patient population we are trying to address? What is the, where in that patient population is there really an unmet need that's not being addressed by other therapies? What is the pathophysiology behind that unmet need? What is the mechanism of action that could tune that pathophysiology? What is the effect of the cell potency-wise that it would have to have when it gets in there to effectively tune it? You might have all the perfect markers biologically, but that doesn't necessarily mean you're going to have potency. And so I think really asking those questions early, and then as we're designing the therapies, designing that up not only for biological, success, but this potency success at the same time is critical. And I think something that that not everybody is focused on enough at the early stages. And I'd like to see more of that. I think that will help us. Once we get to really having a clear goal and a path to get there, then I think in addition to the elements that Eddie mentioned, another one to add to the mix is durability. How long will the effect last, whether it's cells that are supposed to be living or cells that are supposed to modulate the host cells, and then they carry on that effect, versus a gene modification. Is it permanent? Is it temporary? How is this going to have a durability of effect, and what is the desired durability? Permanent, multiple treatments, and how do we really demonstrate that and get to the right effect? And then you said we'll talk about manufacturing later, so
yeah,
I'll hold on that one.
That's a big topic. But Shannon, really, thank you. What I'm really hearing is like you start with the problem, and then you backtrack and then you forward track, not backtrack, forward track what the solution should be, instead of starting with a glorified biological success and then trying to find out what the problem is. It's like I think a lot of us the scientists like to hold on to our academic achievement while ignoring what the actual problem is. It's like holding a hammer while looking for a nail, when in fact you should really identify where the nails are, and then you see if what the right hammers should be for the right kind of nail. So yeah, really, really important point. We have a question from the audience. So from Mark. Mark, I don't know if you are online, if you want to unmute yourself and just ask the question yourself.
Yeah, I'll give it a try. So basically, there are a lot of diseases that are autoimmune that people don't understand. And there's even a lot of funding for stem cell research in California got refunded in addition to NIH. So what's the chance of resetting the immune system when you have autoimmune diseases, or there's really peculiar things going on, like long COVID in some cases looks similar to a known disease called myalgic encephalomyelitis, where your immune system's very messed up, cytokines are weird. I think for long COVID, IL-6, well, anyway, there could be a lot of cytokine issues, natural killer cell issues. What's the chance of resetting your immune system using stem cell, hopefully without having to totally nuke your immune system, but resetting it on the fly versus making you totally immune compromised for a bit first?
Yeah, I'll give this question to Shannon first.
Well, I think I think we're at the cusp of thinking about these issues, Mark. I'm excited personally to see a number of companies that have just emerged in the past year or so around the innate immune system. I think an autoimmune foci. I think this is the next space, kind of following the adaptive immune emergence. So we're not, we're in the early stages of developing that as a field, I think, but I'm excited that it's a place that we're headed.
Yeah, just for perspective, I have a cousin who had an illness. They couldn't treat it in the US. I think it was actually a blastoma or something. And they said he's going to die. So they went to Italy, and they totally reset his, they basically gave him radiation therapy or something, totally reset his immune system. Perfectly fine. US wrote him off. Italy saved his life. So I hope we could do more of that.
We see that also with some MSC cell therapies that have been in development for a while as well. So yeah, I think this is an area where we will continue to grow.
Thanks.
Thank you. Yes, Eddie.
Yeah. Thanks for the question, Mark. Yeah, I agree with Shannon that I was thinking about something that she said in terms of, the immune system is incredibly complex. In some ways, I think the field has turned to cancer because a lot of the recent techniques that we now have for next generation sequencing, single cell kind of transcriptomics and proteomics, it's pretty easy in a sense to kind of go out and kind of grab all of this data about cancer. And yeah, there's just been tremendous investment and interest in that space. So that's making kind of to the point of going out and finding out what the problems are, kind of measuring and characterizing everything, and then coming up with solutions. That's a little bit more straightforward to kind of come up with these hypotheses of how do you effectively go in there and restore a healthy condition. I think in the cases that Mark is talking about, these are really kind of complex imbalances in the immune system. And I think I'm optimistic that as we kind of start to apply these new technological platforms towards understanding those conditions better, I think the engineering is going to catch up, and we can do things that are far short of just nuking the patient's immune system and rebuilding from scratch. There can be kind of much finer ways of kind of coming in with the appropriate scalpel, so to speak, and resetting. And as an example, one of the collaborations that I worked on to get done at Senti between our company and Blue Rock Therapeutics, which is a regenerative medicine focused player that Pear purchased a couple of years ago. They're interested in working with our company to try to create these stem cell derived, basically immune cells, innate immune cells that are endowed with the capability to sense, markers of inflammation. And then, we can actually build these circuits, so to speak, and have the cells kind of respond to that marker, and then, basically kind of correct the imbalance, and kind of, actually put out the right cytokine or combination of them. But the challenge, I would say, is not really doing that. The engineering part is pretty easy. It's like, what do you actually sense? What do you actually spit out? Is kind of the unknown part.
Thank you, Mark, for the question. Thank you, Shannon and Eddie, for the great answer. We also have a question from the audience. Bill, if you want to unmute yourself and ask the question.
Yeah, this is following on Shannon's remarks about potency success. And, there's ways to sort of assess, maybe other new ways to assess potency. There's nanoparticles that we can put in to see if cells are alive or dead or secreting or moving, or and I'm just curious, I guess the broad question is, what are the technologies that you're tracking that might change that practice to sort of give us that potency success? And I sort of follow up on the whole process, the whole workflow of starting cells. There's new ways to edit cells, bulk editing, all sorts of things that people are squeeze and transposons. And I'm just curious what things you guys are tracking that are exciting for you.
I can start with the potency side. So, Bill, thanks for your question. I think it's a combination of factors. What you mentioned with labeling the cells in a sense to see where they're going, I think that shows biodistribution. Once you implant the cells, where do they land? And obviously they'll be more potent if they collect in a region that's relevant to the disease that you're treating. I also like to think about it as what's the job your cells are trying to do. Are they trying to bind another cell type to trigger killing, see if they can bind and kill, in vitro and in vivo? Are they trying to secrete a factor, look at maybe RNA, gene expression and protein secretion, and then an effect of that, again, in vitro and then in vivo with the cell type of interest on the other side. Are they trying to overexpress something? Yeah, so I think it depends on what the function is, but I'd like to see more of that thinking early in the design and development process to say, not only does the cell have to express this, but the result of expressing that is the ability to then have a certain function.
Thank you, Shannon. Go ahead.
Yeah, that was very well put, Shannon. And, I think from the lens of cell therapies for cancer, for instance, I think of potency in a couple of different ways. So, and some of the technologies that were mentioned, say, gene editing, we can kind of touch on the ways that that's impacted the space. One measure of potency for a cell therapy for cancer is just, can you have, kind of a low number, hopefully a low number of cells that you can administer the patient and still kind of get efficacy while, maintaining safety. And, some of the variables that might impact that, right now, the dependency of getting, kind of an appropriate therapeutic response against the cancer depends on gene modification of the cells. So introducing these chimeric, Frankenstein-like proteins called chimeric antigen receptors that allow the cell to actually go and hunt the cancer. And we're kind of using techniques that have been around for a while and are now kind of relatively well accepted, and there's good regulatory precedent for them. So we're using viral vectors predominantly, modified cousins of HIV, like lentiviruses and retroviruses. And so, if we can kind of improve the efficiency of that and scalability, I think we can kind of generate, more potent products. I think there's a lot embedded in potency that we probably have less of a good handle on, because, gene editing is just kind of a tool to, achieve modification of the genome in a particular way. The better we get at it, that's great. We can make a lot of changes within the genome, but what are the right changes that we need to make to, influence potency? Does it mean, knocking out certain genes that kind of serve as brakes or a checkpoint for the cells that we're inserting? Or does it mean overexpressing certain activating factors? These are some of the types of approaches that are being taken in the field in an attempt to kind of improve potency.
Thank you, Shannon. Thank you, Eddie. We also have a question from Ruby. Ruby, if you want to unmute yourself and ask.
Sure. So one of the concerns of allogeneic cell therapy is immune reaction from the recipient towards donor cells. Can you maybe discuss some of the approaches towards overcoming this immune reaction?
I will give this question to Eddie. Eddie, if you want to first begin by maybe explaining a little bit what's allogeneic cell therapy.
Sure. Yeah, it's a great question. Thanks, Ruby. Yeah, we've bandied these terms about a little bit, but there's this distinction between autologous cell therapies and allogeneic cell therapies, and the distinction is, in the case of autologous, you're actually taking cells from the patient and then doing some type of manipulation on the bench or in the lab, hopefully, and then bringing them back to the patient. So it's actually like, from a processing perspective, you're doing that kind of on a single lot basis. It's not kind of a big batch of doses that you're generating. And then allogeneic, just means basically donor derived. So, you can start with a healthy volunteer who may donate cells, whether peripheral, could come from peripheral blood in the case of T cells. There could be other sources. You can start with a stem cell source. And so, yeah, so that's kind of the prospect of allogeneic cell therapy, of course. It's been, hugely exciting in the cell therapy for cancer space, because the first generation of products are all autologous. And there's a number of different approaches to try to make allogeneic cell therapies a success. I think Shannon can probably comment on her experience from, the regenerative medicine side, where, there's also kind of decades of experience to kind of draw on. And so in the cancer cell side of things, there are a few different ways to kind of minimize this problem of, you're using someone else's cell. How do you make sure that the patient's immune system doesn't shut down and you know, kill all of the cells. And one approach actually just relies on taking cells that, you know, I think there's different fields kind of bandied about. They're in a way kind of like immune privileged. So certain types of cells like natural killer cells and mesenchymal stem cells tend to express low degrees of markers that may then kind of result in rapid identification and clearance by the recipient's immune system. So that's kind of like one approach, and that is like relatively well validated, but maybe not as well understood. There's a second approach, which is actually actively trying to make edits to the donor-derived cells to try to make them compatible. And you know, those involve knockouts of certain genes within you know, the cell's genome. So you can use a gene editing approach to do that. And there are a couple of other kind of, you know, I won't get kind of into the details, but a couple other ways for doing that. And then I would say there's probably a third approach of you can actually just match, like, you know, you can now characterize, you know, what are the cells of the donor, what are the cells of the recipient. And I mean, this is often used in stem cell transplants. And, you know, some people are considering these types of approaches in the cell therapy for cancer space as well. But it involves kind of having to bank a lot of different types of allogeneic cells and then having them ready to be administered to the patient.
Thank you, Eddie, and thank you, Shannon. We have a question that's along a different flavor, but equally, if not even more important than the hardcore scientific questions. Afsana, do you want to unmute yourself and ask?
Yes, sure. And I apologize. I think it's you know, it's wonderful to hear about how these therapies are being developed, the fact that they can be life-saving or really preserve or restore function. And so I do apologize for taking this to the angle of cost, but cost comes into play in two ways, right? One is obviously so that people can have access to a therapy that works, a cell therapy that works. And the second are the people that have to pay for it. You know, in real life, in my last job, I worked at a predictive analytics company, and I had an insurance client who asked us to try to predict, you know, how many members in their population would have, you know, CAR T or expensive cell therapies, mainly so they could predict and plan around the spend, because they were really fearful of getting those sudden $1 million claims. So I just wanted to pose the question, how are these, you know, therapies priced? One, so that people can actually have access to them, and second, so that the system can manage around them.
Yeah. Shannon, do you want to take this question?
Sure. There's a lot of great layers to that question, Afsana. You know, it is true, just like we have to think about potency early, we have to think about commercial potential early as well. And really pick patient populations where it matters and think about the impact of actually being able to commercialize these products and fit into the system of healthcare. You know, I think the field is thinking about it and struggling with this issue in real time, and kind of coming at it from a couple of different ways. One, I think, you know, this is really a focus on value-based care, not really fee-for-service, and so the field at large is focused on that angle, and I think the payers are, you know, over the past few years have been evolving to think in that way as well, which is great, you know, timing for this field. People are thinking about it in big companies sometimes as, you know, pay for success, right? If it functions, pay for it. That's more challenging for small companies who might not have the, you know, the resources to support that. So that's an issue. You know, there's other ways to think about it, like how can we really be thoughtful about minimizing the COGS, the cost of goods, for making the product so that it isn't as expensive as well. And that's something we all have to be mindful of. And that's why the field is really interested in moving more and more to allogeneic. You know, the early cord blood transplants actually are allogeneic, right? There's the opportunity with allogeneic for more recent types of cell therapy is it's not one donor into one patient, but now we become a platform where a certain cell source or cell bank can be expanded with great expansion potential. And then the products can be manufactured at larger scale so we can reduce the cost of goods. And so getting to that with the scientific challenges of knowing how to either choose a cell source that's less immunogenic or edit a cell source that's more prone to an immune response are issues. But if we can scale, then we can also simultaneously reduce the cost of goods, which ultimately we'll need to get to for the payer perspective. And so I think, we just like we're staging the development of the science with autologous, allogeneic, adaptive immune system, innate immune system, the gene editing, the gene therapy, all these things. I think we have to look at the payer side with the lens of is there really a good value proposition early on? When we treat this particular patient population with this particular potency, how will we be changed? And with this particular durability and effect, how will that then change the course of care for the patients? And is there really a value proposition there? So we have to be thoughtful about that.
Eddie, do you have any thoughts?
Yeah, no, I largely agree. This is a huge question. I think at the end of the day, as Shannon says, it kind of comes down to what value is being created for the patient. And that's ultimately kind of the standard by which you're going to establish kind of a fair, financial burden that you're going to impose kind of on our healthcare system. So yeah, I think the burden is on the field to try to come up with ways to minimize that. And some of it can kind of come down to creative, financial engineering, like offering a refund in a sense if the therapy doesn't actually end up working. We've seen that. But I think a lot of it can kind of come down to the science of, can we, be more efficient with our manufacturing approaches? Can we use off-the-shelf cell types? And yeah, I'm optimistic kind of about all of these various approaches going forward. And I think we've definitely seen examples where there's been pushback of cell therapies that have not proven out their value. For instance, in Europe and in other places, they're not getting a broad kind of coverage.
Thank you, Eddie. Thank you. Sorry, go ahead.
Try to be mindful, like that. We have to land in a place that's kind of fair for everybody, right? It has to be fair to the patient that we're really changing their outcome and they can get it and afford it. It has to be fair to the payers, it has to work in their framework. It has to be fair to the manufacturers. They have to be able to keep their doors open and keep making the product. And so, it can't be a one angle solution. We really have to think about it with these multiple hats to land in a new space collaboratively so that that we really can make it work for all parties. If it doesn't work for one of the parties, then it won't work.
Absolutely. And I think finding alignment in the values among different in the value chains among different parties is a big topic. It's in cell therapy, in cancer, and also in rare diseases in general. That's definitely a topic that we should discuss, not here, but in a separate panel. But I think all signs point to the fact that we should maybe talk about manufacturing. There is a request from Michelle that we should she would love to hear your perspectives on manufacturing difficulties, and this is also one of the questions I prepared. So why don't we talk about some of the major pain points for cell therapy manufacture during the scale-up process? I don't know, Eddie, if you want to kick it off.
Sure. Yeah, I mean, in my view, it's just been a tremendous crunch in terms of, you know, so many companies, universities, et cetera, you know, getting into the space, wanting to take exciting preclinical stage programs into the clinic. You know, you do have some recently approved cell therapies where, you know, actually this past spring, there was a new cell therapy, a BCMA targeted autologous CAR T product that got approved for multiple myeloma, which was an exciting development. But, you know, BMS, you know, was a little bit embarrassed, I would say, about kind of the fact that the demand was far exceeding supply and that they were having issues, you know, with the process and also, you know, sourcing some of the critical supply, you know, components, which in this case, you know, happened to be one that is a pain point for a lot of cell therapies, the viral vector that's used for gene modification. And then, you know, it happens to be the case that these cell therapies and interest in them is exploding at the same time that there's a lot of interest in gene therapies, and a lot of the facilities that are, you know, and CDMOs that offer these services, they're trying to keep up with the demand. But yeah, you know, it's going to be, I think, a pain point for a while. A lot of biotech companies, like, you know, the one that I'm at, you know, we face this decision of, do we, you know, kind of go with a CDMO and you know, buy supply, and there's a number of challenges kind of intrinsic to that, or, you know, kind of going on and building our own facility at, you know, with all the investments, you know, that comes therein, or, you know, some type of hybrid approach depending on parts of the process. So, you know, at Senti, we're doing a little bit more of the hybrid process, but we are, you know, recently, it's been very exciting that we've gotten committed to building our own manufacturing facility. And actually earlier this week, I was at a groundbreaking for our new facility. We signed a 92,000 square foot facility lease in Alameda. And yeah, it's amazing just, you know, what this kind of represents in terms of a commitment of, you know, over the next year, we're going to build out this facility with GMP manufacturing suites. We're going to hire, you know, dozens of employees to support it. And it, you know, for us, it signals confidence in our programs that we hope, you know, we're going to be producing, you know, dozens of doses, but it's not without risk. And it's certainly incredibly hard to kind of find the talent to, you know, run this facility. You know, I read an article about the, you know, some of the insane job offers that went out to people who are, you know, one of the few who have expertise in this particular space of, you know, CMC process development, analytical, you know, development for cell therapies. So I think this condition is going to be around for a little while until kind of the supply catches up with demand. And there's a lot on the engineering side that can be done to kind of improve the process from a scalability standpoint.
Yeah, absolutely. And Shannon, any last quick thoughts? I know we're a little bit over time.
Yeah, I agree with all those things that Eddie said, and some key business choices there. I guess I'll add three more things: science, scale, and funding tension. So the science, what are you trying to make? And often this gets staged. So you might make something at small scale and then you think, oh, then I'll just make it bigger scale. But actually in our space, the process is the product, and the way in which you make it is often different at scale and can cause risk. So that's, you know, I think the science and the science of manufacturing really have to be figured out in parallel very collaboratively early on. In terms of scale, I think we're at an exciting place. You know, when I started early on, we didn't have bioreactors that could be used for cells. You know, people were using roller bottles and like these other things. And, you know, we had to develop a lot of our own bioreactor technology. So it's very exciting to see, you know, at least there's now available tools and systems that can support scale of manufacturing, both both autologous and allogeneic, and I think we're seeing more and more of that come online. So like we have the tools of gene editing, we have the tools of production, but the science, it's not just, how does it work in those new tools? It's also like, how do all the little choices you make along the way, spanning from expansion potential and ingredients that you put into the media, et cetera, and things that you edit in the cells purposely or non-purposely, how do they land? So pairing those together is critical. And then the last one is that funding tension, because it is, I think for long-term success, it's critical to pair that early, but sometimes the funding isn't always there. And I've heard this is true in all companies of all sizes, right? It doesn't really matter. It's kind of this sense of, let's get the proof of concept data first, and then we'll invest. But I think this space is particularly important to know that the proof of concept is achievable with the scalable process and the final process. And even FDA requires that before you go into the clinic, you at least have to lock in your materials for scale. And so kind of, having the science and the manufacturing and the funding all together on the same team is, I think, going to be, always critical.
Thank you. And I think really that speaks to the multifaceted aspect of cell therapy. That's not just a scientific challenge, but also a logistic challenge, a legal challenge, an ethic challenge, and also a cost, a challenge on cost and aligning value system. So I know I'm sure there are more questions that the audience would like to ask, but we are out of time. So thank you very much for all of you to be here, and thanks for the esteemed panelists, Shannon and Eddie. And if you are interested in engaging further with us, I just again put in the chat a link for an info session on a conference that I'm leading the organizational effort of. It's your engagement is very much customizable. You can work for as much as you want or as little as you want, and it's a great way to broaden your network and chat with like-minded folks like the ones that you're seeing right now. So with that, in the event, I did promise a little networking session at the end. So first of all, let me stop recording.